Hello, everybody, and thank you for joining me in this discussion today. My name is Olivia Achikasim, and I'm a junior biology student at Oglethorpe University. And today I will be talking to you about cell-to-cell communication through tunneling nanotubes. The reason I decided to focus on this is because right now I'm taking a developmental biology class or course, and early on in the semester, we discussed cell adhesion and how important it is for all cells to kind of know what is going on in order to adhere. And by adhere, I mean stick, the primary definition. And I wanted to see how cells manage to communicate because I'm very sure they do not have mouth to speak. And before now, I thought that they all did their own thing and somehow everything managed to work together for the good and make everything work at the same time. So moving on to the objectives. Today, through this presentation, I hope to help you understand why cell-to-cell communication is important. And I'm going to be discussing mechanisms of cell communication. We'll be understanding tunneling nanotubes, the formation, the structure, the architecture, what makes up the nanotube, and the function. We're also going to talk about how it transfers molecules and organelles through it, and you watch a video on this too. We're also going to look at experimental studies that prove the presence and the relevance of nanotubes. Another thing not on this slide is that we're going to look at the application and the implication of this novel mechanism to biology. To help us understand how cell-to-cell communication works, I want you to think back to elementary school days. In particular, I want you to think about the things you do in order to secretly talk to your friends during class. So here you are sitting in classroom and you desperately want to tell your friend something. So you write a little note and you pass it to them. When you pass it to them from your hand directly to their own hand, they see the note and no one else does. It's just them seeing the note. But let's say your friend is across the room, and I don't know about you, but when I was in elementary school, I would write notes on a paper and fold the paper up into a paper airplane. And I had pretty good aim back then. So I'd write my note and throw my paper airplane over to my friend across a distance. And again, they would be the only one to get a message, even though I threw it across a distance. So if I wanted to tell a few friends or a group of friends something, I might call a little huddle and say a few things just to the group of friends, and my voice will only have to go through the little distance between us. Now, I had a classmate in high school who was a rebel, and sometimes he would go over to the secretary's desk and take over the intercom to say funny things, maybe to say things like, meet me at the flagpole or on the playground or in the bathroom during recess. And I'm sure all of us know this how the intercom worked. So this intercom message will go out to everyone and be broadcasted to the whole school. Those who wanted to meet him would, and those who wouldn't want to meet him just wouldn't go. So think of cells as little people that do similar things because you might not think about it, but it is important for cells to talk to each other, to communicate with each other. So evolutionarily, cells being able to communicate with each other is the major reason why we as human beings are so complex. So how do cells pass notes to one another? How do they communicate directly with each other? One is by direct contact. So that's when a cell touches a cell, which is direct cell to which is direct cell-to-cell communication. And they also communicate over short distances too, which would be our paper airplane, because our paper airplane is traveling over a short distance, which will also be me talking in the little huddle and the voice is going over a distance. Either way is going over a distance. And a biological example would be a neuron releasing a neurotransmitter in order to communicate with another neuron, and it would diffuse across a distance. As I mentioned when I spoke on direct communications, neighboring cells, that cells close to each other, communicate through direct contact. So what about those that have quite some distance or even a lot of distance between them? This is where tunneling nanotubes come in. So throughout the lecture, you might hear me sometimes call tunneling nanotubes TNTs. It's also tunneling nanotubes, so take note of that. So tunneling nanotubes are long tube-like structure. So think of it as, when I read this, I thought of, okay, PVC, the tube, or the tube in your bathroom or something. Just think of a normal tube. Don't think too far. So tunneling nanotubes are tube-like structures, and they were first identified in 2004. And you know, the funny thing is that they do not contain microtubules, which I found to be really ironic, because when I think microtubules, I think movement. Yeah. Another cool thing about this tunneling nanotubes is that they are sensitive to stress. So during the experiment we're going to talk about later on, the researchers observed that if they move the petri dish with the cells in it too much, the nanotubes ruptured and just destroyed. In this paper, the researchers sought to prove that this novel mechanism, that's the nanotube, is good, and it's actually really good at what it does in cell-to-cell communication. So the reason I put this up here is in case you wanted to find the paper. There are two papers with this topic, so but they don't have the same front page, and that's the reason why I put this on here. So the methods. The researchers obtained PC12 cells. So PC12 cells are obtained from a rat's adrenal medulla, which forms from the neural crest, and they got this because it's easy to culture. So the researchers cultured this PC12 cells, and then they performed a 3D live cell microscopy in the presence of fluorescently labeled wheat germ agglutinin. So wheat germ agglutinin separates the plasma membrane from glycocalyx, GLYOCALYX, which is what covers the plasma membrane, and they observed the structures of the cell. So on the next slide, we'll be watching a video of this transfer between the nanotubes. So before then, this image on my right is what the PC12 cell looks like when cultured. It has different morphologies. That's why I put all the images up here. But this is what a PC12 cell looks like. Now on the next slide, we're going to be watching a video of transfer through the nanotube. I'll let you watch the video to the end so you identify the nanotube on your own, and then I talk about it. It's not going to be really difficult to identify, seeing I've introduced like the tube-like. So just look for tube-like, and then I'll talk about it. This is after a few days of watching and observing the cell. It's a PC-12 cell too. I'm going to replay it again, and this time I'll talk over it for those that couldn't find the nanotube. So this right here, where my mouse is the nanotube. And the little black things you see moving are the molecules and the organelles that are going through the nanotube. A cool thing I learned is that a nanotube also transfers mitochondria, which I thought was like, this is new information because I didn't know you could transfer mitochondria. In fact, I thought the mitochondria was like DNA, unique and only one, like it's irreplaceable. That's what I thought. So yes, this is the nanotube. These are the cells. These are the cells. These things that you see extending, let me take it back a little bit. So these things you see extending are the philopodia. So, sorry, this is the philopodia. So the first to get there is the one that's going to be the nanotube. This is the nanotube. This is the cell. This is the cell. This is what it's transferring. It's going, and has gone into the other cell. Yes, so that's that. That will help you understand everything else I'm going to say. So this is an image from the paper that it's showing the connectivity of the tunneling nanotubes within cells. So I don't know if I mentioned it earlier, but the way nanotubes are formed is that these things that are like cilia, it's not cilia, I'm just giving an analogy, but these things that are like cilia extend from the cell. It's called philopodia. Multiple philopodium extends from the cell. And the first philopodia, I'm sorry, anyone that knows the plural and the singular should please correct me in the comments. The first philopodium to get to the other cell is going to be the nanotube. So the others that are trying to reach the cell begin to degenerate. I think of, when I see this, I think of sperm and egg. The same way one winning sperm gets to the egg, and then the egg just blocks out the rest, and the other sperms take the L. That's the same way it is with the nanotubes. So all this image is showing is the connectivity. So as if you look closely in C, we see adjacent connections between cells. That you see this cell over here has a nanotube that's connecting all the way to this cell over here. And that cell is connecting to multiple cells with multiple nanotubes. And in B2, you see the multiple nanotubes to multiple cells. And in D, one nanotube, one cell. A and D are kind of like similar. The reason I put F here is that if you look closely at the edges of the nanotube where my mouse is, that edge, well, you see clearly in that edge. But if you look closely, you see that the nanotube fuses into the cell like it's not something that was formed outside. It fuses into the cell, like that's how it has been, connecting these two cells together. So another way I think of this is like the fusion between the nanotube and the cell is that I think of this as like the carbon bonds that we learned about in chemistry or organic chemistry. So if you've taken organic chemistry, you know that when carbon and carbon bonds. The bond connecting the first carbon to the second carbon is a bond that's totally different from the one connecting the second carbon to the third carbon. So I'm going to draw it up here. So this is carbon 1 with the bond connecting to carbon 2 with another bond connecting to carbon 3. So the let me call it cell 1, nanotube, cell 2, nanotube, cell 3. So the nanotube connecting cell 1 to cell 2 is different from the nanotube connecting cell 2 to cell 3. That's how nanotubes bind. So it's not, the nanotube connecting 1 to 2 is not going to be the same nanotube connecting to 3. In fact, the nanotube connecting cell 1, if say there's a nanotube that's meant to connect, let me draw that. Say there's a nanotube that is meant to connect C all the way to 3. That nanotube is going to be different from the one. That nanotube is going to be different from the one connecting cell 1 to cell 2. So yes, that is it. No, I went ahead. Okay, something important to know with nanotube is that actin, ACTIN, plays a major role, a primary role. It's like ingredient number 1 in the formation of nanotube. So here, the researchers preloaded one cell with actin and myosin and preloaded another cell with only actin. And with this, they wanted to see if the transfer will occur. So I don't understand the reason why D doesn't have any color. I do not know. But in the first portion, in the first portion of this thing, that's A, the cell in green is the cell that has actin plus myosin, and the cell in blue is the cell that has only actin. So they wanted to see if the cell with actin and myosin will transfer myosin to the cell that only has actin in it. So here, where the arrow is, I'm sure you can already see the nanotube is already doing its thing and transferring the myosin to the actin-only cell. And in D, though it doesn't have color, you can see that where my mouse is, that the myosin is left, and through the nanotube, the cell the cell that had actin and myosin has transferred its myosin, even though it's like sacrificed itself and transferred almost all the myosin it has to cell B. So that's that. And there's another image on the next slide that's going to make this clear a little better. It does justice to this. Well, does it really do justice? So here, this box over here, this is the gray version. Here, this is the nanotube, and here is the nanotube with the cells around. And here is the cell. And here is the nanotube. The one with the box around it is the nanotube, and the cell is at the other end. And in this nanotube, where this fluorescently exposed molecules are, we see that the nanotube is carrying something in it, which I assume is going to be transferred to the, I'm sorry, I have a little cold, that's why I keep stopping, which I assume is going to be transferred to the other cell. Yes, it's going to be transferred to the other cell. So, oh, I'm sorry, I didn't know my illustration was still up here. Let me erase that quickly. Okay, so in this slide, the researchers performed another experiment because as scientists, they replicate this experiment multiple times to confirm if it is what it is. So here, the researchers performed the experiment again in mesothelioma cells because with the previous experiment, that's in the PC12 cells, the PC12 cells are kind of like round cells. So you don't really understand the orientation of the nanotube. So, and these mesothelioma cells are flat cells. So you're able to understand the orientation and the structure of the nanotube, and it's flat, and it's easy to, it's just easy to visualize. So here, the question they asked in this one is, what is the molecular composition of this nanotube? And does this nanotube, or do these nanotubes have cytoskeletal components in the structure that actually make the thing function? And that's what we're going to be looking at. After their culture and everything, and the method, they dyed or they stained this with DAPI. Okay, yeah, you can see it up here. And everything on this slide in the image is what they found in the nanotube. Though the concentration of some was higher than in others, but I'm not going to go through all of them so I don't bore you with detail, but I'll go through most of them. So in B, slide B, or figure in B, image B, fasten. I don't know if that's how they pronounce it, but that's what I'm going to call it. Fasten was found in the nanotube, and fasten helps facilitate the cell extension by cross-linking the actin filament in the TNT in the tunneling nanotube that extends to the cell. So it's what makes the TNT extend to the other one. And Esrin mediates, yes, it allows for anchoring of the actin microfilaments to the cell membrane. It allows the nanotube to be able to latch on to the cell membrane of its target cell. And now E-cadherin, the result for E-cadherin was special because the researchers wrote in the study that they did have a lot of struggle identifying the E-cadherin, and when they did, they saw that E-cadherin was minimally expressed, but then they think it's because maybe they had a lot of problem identifying this, or it just wasn't formed a lot. But they had another study they performed to see if E-cadherin was there, but that's outside the study. So here in this place in red is like E-cadherin is there, but it's not there, but it's there, but it's not there, kind of thing. That's why I circled it. And for ZO1, oh, I'm so sorry. And for ZO1, for frame G, ZO1 links the ends of this tunneling nanotubes to form tight junctions that allow for cell-to-cell contact. Now, this is where my takeaway slide begins, because I'm trying to remind you of how tunneling nanotubes are formed. So there's cell dislodgement another way, but that's not what the focus of this presentation is. My presentation is focused on actin-driven protrusion of the nanotubes. So here you see cell 1, where my mouse is, you see cell 1 and you see cell 2, and the nanotube is formed basically in 3 ways. So in cell 1, cell A, we'll call it cell A and cell B. So cell A could be the one extending the philopodia to the other cell. And when the philopodia from cell A touches cell B, the nanotube is formed, like you see here in the arrow after that. And you also see that, say, cell A is extending its philopodia and cell B is extending its philopodia. When those philopodias, someone please correct me, I know I'm wrong. When those philopodia meet, it fuses together to form the nanotube, like we see in at the right. And then C is similar to, the third model is similar to the first, just that. You know how I told you, well, let me go back to the previous slide. You know how I told you this forms tight junctions? So here, the green thing is like a little space left between, which I call the breathing space. It's like, if you're an introvert, you know, okay, sometimes I want to be here, but sometimes I need to think. So it's like, I call that green thing like the thinking space, the breathing space. So it's like the nanotube extends, but it doesn't like latch on to the cell membrane. It latches lightly to the cell membrane. So that's what is here. And then this is the example I gave you of the carbon-carbon linking, where the nanotube connecting the first carbon to the second carbon is not the same nanotube that's connecting the second carbon to the third carbon. So it's if cell 1 or carbon 1 wants to attach to carbon 3, another tube, which I'm going to draw on here, another tube has to come all the way around and latch on to carbon number 3. So that's it. And then, yes, that's it. Now the takeaway slide. I want to remind you of why cell communication is important, because for you to actually, or for the cells to grow, cell communication has to happen. And your brain cells cannot be thinking one thing and your hand cells are thinking another thing. They have to, basically the way when we work together on group projects, we all want to know what's going on, so we know how to move forward, so that if we have to hold someone accountable, we know who to hold. It's the same way cell to cell communication works. It's so important to work. And the nanotubes are built primarily of actin, and it causes this fusion between the cells that it makes the nanotube seem like, oh, I was meant to be there, that kind of thing. It just fuses into the cells it's connecting. And now the relevance of nanotube to biology. A major thing I found to be really interesting is that nanotubes can be used to fight diseases. For example, nanoparticles can be moved through nanotubes, and the nanoparticles can be loaded with specific drugs that will go to the nanotube, which is so cool. And then I also saw that it can also, while it's transferring drugs through the nanotube, it can also, if say cells that diseases that are passed from cell to cell, because the drug is moving from cell to cell, the treatment is moving from cell to cell, and it's fighting off the diseases from cell to cell. And 3 is that the nanotube allows for preferential uptake of mitochondria to increase survival and for better resistance to stress. So, and this here below, I'm sorry, I'll move my video up a little. This cartoon below is a little fun cartoon I made to remind you in a fun way uh, what cell-to-cell communication is and why it's important. This is a summarized summary and a fun one I think each of you will be able to remember. So thank you. That's my presentation on cell-to-cell communication. And once again, my name is Olivia Achikasim, and I'm a Oglethorpe biology major student. Thank you for listening to me.